emerging at 6.7 billion and on its way to reaching 9 billion by 2050, the world's food production will probably need to double. So between the world's expanding population and the current global efforts, climate change's effort to grow more food will be inhibited by water shortage. You might wonder how well you and I only drink about a quarter to water a day, but in the same day, the food Americans eat require 2,000 to 3 quarts of water to produce it. When farmers experience poor growing conditions, their crops can be reduced by up to 70% and more around the world. The amount of land affected by drought has doubled in the past 30 years. We need alternatives and fast that where the biotechnology is breaking through. Again, biotech crops that are drought resistant may play an important role in enhancing food production. So how is that the possible? Well, during drought, plant naturally drop leaves to favor the roots. That's not the best in agriculture response, since the plant will then not produce. In order to self-preserve, one way to help avoid this is by using what are called transcription factors, which are like master regulators. They are able to turn on dozens of other genes in the plant, help to orchestrate its response to lack of water. For example, instead of gulping water, the plant could just sip water, so we can get more crop per drops.
Plants are photosynthetic organisms able to feed atmospheric carbon and macromolecules essential for growth and survival. Thus, carbon metabolism and accumulation are highly regulated. This regulation has a vital role in plant resilience to stress and crop yield. In maize, the catabolic enzyme T6P-phosphate or TPP has been specifically expressed in female floral components using the rice floral promoter gene MAD-acid. This reduces the concentration of T6P in female reproductive tissue and increase the sucrose level in a whole developing spikelet. It also affects the T6P regulatory pathway. Effect on drought resistance were assessed in field and the yield was improved in well-watered, mild and severe drought conditions, with no obvious impact on plant. As plants are impermeable to exogenous T6P, synthetic precursors were produced and used as treatment that trigger a light-inducible endogenous production of T6P. Tree hallux was also shown to accumulate in the roots of plants and augmented brassinosterol or BR signaling together with other osmoprotective sugars like sucrose and raffinose. Furthermore, T6P-related gene expression was specifically upregulated in the root flower cell. In turn, by mediating BR signaling, sugar act as signaling molecule in Arabidopsis to control different aspects of fruit system architecture, such as primary root elongation, lateral root development, and root directional response. The manipulation of BR signaling results in an increase in osmoprotected metabolites, including proline, which is an amino acid long known for conferring drought and salinity tolerance. Thus, accumulation of sugars and proline might also be a valid strategy to achieve dehydration tolerance in cereal, as shown by transgenic maize plant.
The challenge and future perspective in these issues are genome editing for drought-resistant crop and tissue-specific promoters to drive drought tolerance. Firstly, genome editing technologies like zinc-fingered nucleases (ZFNS), transcriptome activator like effector nucleases, and homing meganucleases are common methods that scientists use to produce targeted genetic modifications in the organism. So, as the challenges still need more time-consuming and labor-intensive. Besides that, it also gives challenges in genome editing for drought-resistant crops, as it still have limits to genome editing in crops for plant transformation efficiency, which hampers the delivery of genome editing material into the target cells. Next, for the tissue-specific promoters to drive drought tolerance in this research, it gives challenges in limited availability of promoters that allow such specific gene expressions, but it could be overcome by performing transcriptomics in crops under normal and stress conditions that accurately differentiate between tissues. So, hopefully, it can lead to the isolation of tissue-specific drought-responsive promoters. For the future perspective in these issues, as sufficient numbers of promoters are identified and tested in the crops, a visual cycle might be triggered in which transgenic series expressing tissue-specific markers would enable the tissue-specific transcriptomics. In addition, this cycle can lead to the discovery of novel cell type and response-specific promoters that might provide innovative solutions to plant biotechnologies.
Assalamualaikum. Okay, I will continue the presentation. For the conclusion, the genetically modification organism can help in the food production, drought resistance, climate change mitigation, and the disease resistance. But in addition, it may produce number of threats in the gene, okay, individual, population, and ecosystem level. Okay, they can also have an impact on the agro industry of developing countries. The GMOs can be complementary to the traditional crops, but by no means can they replace the traditional crops. The world must ensure that the standards are followed in using the GMOs and are being continuously monitored. Okay. The developing countries like Pakistan should ensure strong scientific capacity and research based on approach in the field of genetically modified organisms.